Okay, we're gonna get going here. So, bonjour, Anine, and welcome to the final installment of our 2020-2021 GLIER T-RUST virtual seminar series, which is our student-run lightning talks. My name is Corey Bertrand, and I'm out of GLIER in the Heath Lab.
My name is GLIER T-RUST. I'm from the T-RUST program.
And we are your hosts for today's showcase.
To begin, we would like to acknowledge that the University of Windsor and Wayne State University sites on the traditional territory of the three Fire Confederacy of First Nations, Anishinaabeg, Peoria, Miami, and Mississauga Nations.
We, as the Windsor and Wayne University communities, would like to move beyond acknowledgements, and we would like to not only give our respect to the traditional territory we are on, but also commit to learning and growing, starting with our partnerships and collaborations moving forward.
This is the concluding student showcase in the 2020 and 2021 seminar series.
Today, we will be showcasing a series of student projects ranging from our diverse scientific backgrounds. We have a mix of live presentations and pre-recorded presentations.
As a housekeeping, as part of the Zoom ground rule, everyone should already be on the mute and have their video off. Please confirm that, and if not, please mute and turn the video off now.
If you are presenting today at the conclusion of the previous presenter, please request control at the top of your screen under View Options. This will allow you to move your slides forward using your mouse. Caution, there is a delay. Otherwise, if you are comfortable saying next slide, tech support will advance the slides for you. Due to time constraints, we will not be having questions for each individual speaker today. However, we will be requesting that you fill out a unique survey form, which will enter you into a draw for multiple prizes, including 6 memberships to the International Association for Great Lakes Research. Each survey you fill out for a speaker will be an entry into the draw. Lastly, the entire seminar today will be recorded and placed on our GLIER T-RUST YouTube channel. So now with our ground rules set, please help us welcome our GLIER director, Dr. Mike McKay, to give a short introduction.
Thank you, Corey, and good morning and welcome everyone to the 2021 GLIER Trust virtual showcase, an opportunity to come together to celebrate the accomplishments and perseverance of our most valuable asset, our graduate students. So the ongoing COVID-19 pandemic demonstrates the fragility of the world as we know it. Yet it may also serve as an environmental inflection point where lessons learned from our common experience during this crisis may prompt society to take a different path when it comes to environmental stewardship and sustainability. Our response to it underlines the imperative of resilience, flexibility, and finding new means to work together to innovate and solve complex problems, whether they're economic, social, or environmental. And one of the best ways to do so is to bring together the right mix of people and resources in an environment that leverages expertise from a wide range of perspectives. And this series, bringing together diverse voices and experiences from the University of Windsor and Wayne State University, is an excellent example of how to do just that. So congratulations once again to our graduate students. You're an asset to our respective universities in the Great Lakes region, and we look forward to your continued successes. And before leaving, I'd also like to take this opportunity to announce the 2020 Lum Clark Research Excellent Award recipients from GLIER. These awards are offered to GLIER students each year, one at the master's level, one at the PhD level, with each award worth $1,000. It's noteworthy that these awards are adjudicated by your peers, graduate students in the GLIER Environmental Sciences program who volunteered to serve as reviewers, and I'd like to express a big thanks to those who volunteered. So with that, I'm pleased to announce that the 2020 Lum Clark Research Excellence Awards go to 2 recently graduated GLIER students, Sarah LaRocque in the PhD category and Amy Weintz in the master's category. Thank you, and I look forward to this morning's presentations.
Thank you, Dr. McKay. Our next introduction is Dr. Dan Meno, who is our Associate Dean of Science, Graduate Studies, and Research.
Thank you, Corey. Yes, my name is Dan Mennel. I'm the Associate Dean of Science for Graduate Studies and Research at the University of Windsor. I'm also an ecologist, an ornithologist, and someone who values the Great Lakes and environmental research into the Great Lakes in a deep and personal way. When we look back on the stormy year behind us, I think it can be a real challenge to find silver linings. But I really think what you have achieved with the GLIER T-RUST virtual seminar series is a silver lining. You have seized this opportunity and the new technologies that have all become part of our daily lives to build new international relationships. You've come together to share ideas and to share your research perspectives in the environmental sciences. And I think that is something very special. And I think we should find ways to continue this thing that you have built after the pandemic concludes. I think it's especially meaningful that the GLIER T-RUST virtual seminars were spearheaded by graduate students. As Mike has said, you are the heart of research and future investigations into the environment and our impacts on the environment. So on behalf of the Faculty of Science at the University of Windsor, I congratulate you on an excellent year of student-led international seminars, and I look forward to the event ahead of us today focused on the research of graduate students at the University of Windsor and Wayne State University. Thank you.
Thank you, Dr. Manning. On behalf of the T-RUST, please let me introduce Dr. Donna Kashian.
Hi, everyone. Thank you for the remarks from both Mike and Dan. They were wonderful, and as a co-sponsor of this, I reiterate all of that regards to our students and what they've accomplished to this. And on behalf of Wayne State University, as the Director of the Environmental Science and T-RUST program, I want to welcome and first congratulate the students on this accomplishment of organizing, running every aspect of the joint seminar series this past year. If you could have been on this Zoom with us for the last half hour, watching them coordinate and put together a technically rather difficult presentation for you today with multiple talks, some of them recorded, some of them not. They have mastered this, and they've operated as a family between the two universities. It has been wonderful. I would also like to welcome all the guests that have joined us throughout the semester and that have joined us today. For those that don't know much about T-RUST, it's an innovative graduate training program at Wayne State that integrates both social, physical, technology, engineering, mathematics in an effort to tackle the complex challenges in urban sustainability. One of the things in which is the foundation of the seminar series. Its goal is to train our graduate students, a diverse group of graduate students, to integrate scientific engineering, social scientific approaches to combine innovation policy and foster innovation of urban sustainability. Again, another thing that is highlighted by this seminar series. So in collaboration with GLIER, this represents what can be accomplished with these partnerships and bring in a diverse group of voices to address issues in climate change, traditional knowledge, industry, on and on and on, and everything we're going to hear from the wonderful students today. And it's with the students' ideas that came up with the topics for all of these. And they went on to take what is typically a traditional seminar series with one speaker to integrate these panel discussions and leave at the end of each one of the seminars and panels a bit of time at the end for a more intimate conversation regarding careers and professional development with the speakers, something very unique to the seminar series. With this, I want to thank all the students. You've been amazing and wonderful, and everyone who's attended this. And I would like to mention that one of the co-hosts for this is also Dr. Scott Mundell at the University of Windsor. He was supposed to give a brief introduction. He is in the field, as every biologist should be in the field, and his connection wasn't good enough to join us today. So please recognize his efforts and contributions to mentoring these students too. And so with that, I would like to pass it on to our third co-host, Dr. Kat Fabreria.
Yeah, thank you, Dr. Kashian. Finally, we would like to introduce Dr. Catherine Fabreria and ask her to share her experiences from the past year.
Thank you so much. What else can I say following all of those comments? I think it really has been a privilege and a wonderful opportunity for Donna to be so open to work with Scott and I to rethink our graduate seminar series. We knew at least since I started teaching the course with Scott in 2019, that we wanted to bolster the partnership across both sides of the river. And Donna and I have been in talks for quite some time around how we can better inspire and connect and train our early career researchers. So this is really an example of that. If Scott were here, he's been texting me in the field in Alberta. He wanted to emphasize just how critically important the opportunity it was to engage with people in the industry and on panels. He's received feedback, and we've all received feedback about how conversations with these early career researchers have continued on beyond the panels, after the panels, between groups and with the experts. So we can definitely see the benefits of this sort of collaboration extending well beyond the series itself. So that really is a wonderful outcome of it. And I'd like to just also recognize how the students have collaborated in this as a scholarship enterprise. We really did think about every aspect of the work. So I'd like to ask Katrina to just put the next slide up. We've collaborated throughout the process around how to do this and whether or not we could use this as a model for other things. And so for those of you that know the 10 Simple Rules series of academic hacks, we decided to co-write one ourselves. So we landed on 10 key ingredients that hopefully you'll notice over the course of the next 2 hours, the outputs of that or outcomes of that, and feel that sort of community that we've built. It started with co-creating a partnership back in August. We defined and discussed a way of working, landed on a unified vision and theme, this idea of transformative change and environmental sustainability as being the threads that connect both the GLIER, which is a more physical and natural science-based program, and T-RUST, which is urban sustainability and a lot more interdisciplinary across social sciences and communications. You will see that reflected not only throughout the series, but in today's research talks. We also challenged ourselves to think about non-traditional structures, topics, and formats. And you'll see that within each talk and across the series, including today, that dimensions of inclusion, diversity, and equity were all embedded in the program and the programming, and in small and large ways. And every role that you see here today has a very clear description, a definition, and that's evolved over time as we learned and grew together. And every single role here is co-led by one GLIER or Windsor student and a T-RUST student. And so what this allowed us is that we could peer mentor one another, and very much, Donna, Scott, and I are very much colleagues with these early career researchers learning this space together. Everyone has had an opportunity to lead, and everyone has had a chance to develop in very small and large ways professionally. We really thought about making it accessible. So not only is this being synchronously shown today, it is going to be recorded, and we have our YouTube channel as part of that. And we've also generated metrics that we've been tracking over time. We've had a minimum of 50 people attending our sessions every time, with some of them being, well over 110 attendees. And that just tells us that we were able to amplify and connect with a global audience we couldn't have done individually in isolation. And then a couple of things that are really my favorite is that we've been able to foster a welcoming environment. We started it sort of mid-series about just giving a wave, and that's been a tradition for this series. And you'll sort of see all of us kind of coming online just to encourage and build a sense of community in this otherwise isolating sort of environment. And today is really point 10. We wanted to showcase and champion our future leaders. And so it is really a pleasure and a privilege for me to step away now and stand back and be muted for the rest of this and really just watch the students soar. So join me in congratulating the group. The next slide just shows you all of the sorts of tools and hacks we have leveraged as much as possible to make sure that we could do this without ever meeting. None of us have ever come together in person. And so, just follow that YouTube channel. It'll be sort of a really nice memory, and hopefully this publication that comes forward from this as as something that we can all reflect on and realize has been a wonderful new way of doing research, and will certainly live on in all of our programs and career pathways moving forward. So thank you very much, and I look forward to the rest of today, and congratulations, everyone.
Thank you, Dr. Fabria. If we can move to the next slide, please. One more. So here's our schedule for today. We are right on time. We've done our opening remarks. We're going to have 3 sessions and then breaks in between.
We will be splitting our speakers into 3 sessions, which include speakers from GLIER and T-RUST. We have 3 minutes for each presentation and 2 10-minute breaks in between each session. When you reach your time limit, you will receive a warning in chat from Corey or me. And now please welcome our first speaker, Corey Bertnant.
Okay, so hello, my name is Corey Bertrand. I'm from GLIER, and Dr. Daniel Heath and Dr. Pedro Perez Neto are my supervisors. My project is called Exploring Seasonal Effects on Riverine Fish Communities using eDNA. I am in the unique position being involved with the GenFish project, but also conducting my own thesis. While these are unique projects, the two are heavily integrated. Every organism has a unique DNA barcode, and for fish species, this DNA is shed from scales, mucus, gills, just to name a few. That DNA becomes suspended in the water and is thus known as eDNA, or environmental DNA. Using a glass fiber filter, we can pass water through the filter, which will capture the DNA, and we can take that and process it back in the lab. Under the umbrella of GenFish, we are looking at producing an eDNA chip, which can be used for all fish species across Canada. Using eDNA to determine what fish are present in a water body, or what is called presence, how many, or abundance, and how they are interacting with one another is one of the main goals of both GenFish and my project. My project seeks to explore the seasonal effects on riverine fish communities using eDNA. This will also, in turn, allow us to explore how eDNA acts in a river setting over the course of a year. To accomplish this, a lot of sampling must take place. Under the GenFish project, we are involved in sampling upwards of 500 water bodies across Canada. Here, we can see what I was able to accomplish last summer. On the left-hand side in yellow, we have the 17 sites along the St. Lawrence that we were able to sample with our collaboration with Odyssey, Quebec. On the right, we see the 29 sites along the Credit River in Mississauga. In the middle, we see the 50 sites of urban Windsor on the southern shore of Lake St. Clair, and also the 30 sites in and around Point Pelee National Park. In total, I was able to collect samples from 119 sites across Ontario. But what do we do with the samples? Before we can even move to the samples, a lot of work must be done to create primers and validate them using a new protocol that I'm a part of creating. Once we can create and validate our primers, we can then test them using our water samples. Looking at the metadata for the water bodies that were tested, we can then compare efficiency of eDNA in terms of presence and abundance to more traditional methods such as netting and electrofishing. To further our knowledge and to provide ecologists, government organizations, the ability to know not only what species are present, but how many are there on a provincial or even national scale. Now that's pretty unique. I plan on developing approximately 30 species-specific primers, which includes invasive and species at risk, to be able to use real-time quantitative PCR to determine abundances and presence of these unique species. This will allow monitoring of our lakes and rivers to maintain and protect where we find high abundances of species at risk, but also to detect and determine invasion points for non-native fish species as well. The main goals of determining presence and abundance using eDNA will allow future stakeholders more options in studying and helping maintain our sustainable fisheries. This and the ability to understand seasonal impacts on fish communities will help us to understand how to protect fish species into the future. Lastly, with the ever-changing climate, eDNA presents itself as a viable option to not only understand environmental stressors on fish, but act as an early warning system against invasive species and to help uncover highly cryptic species as well. In totality, we are creating an additional tool to help in the arsenal of protecting our fish species now and into the future. Thank you.
Thank you, Corey, for the lovely presentation that you have. So now we want to welcome to our next speaker, who is the Madison Doctor. And if Madison, you are ready, we can start.
All right, hello, my name is Madison Dugdale. I am a first-year master's student at GLIER, and I am in the picture lab, and my thesis is looking at suspended sediments and how they affect the red side dace, shown in the picture below, and their swimming performance. So there are multiple stressors that affect fish, such as increased temperature, nutrient input from runoff, as well as suspended sediments, as you can see in the top picture. These stressors, especially sediments, impact fitness and populations of aquatic species at risk because they live in close proximity to areas of increasing urbanization, and also because they are more sensitive to changes in their environment. The species at risk I will be looking at is the red side dace, which is endangered in Canada, and they face multiple threats such as habitat loss, alteration, and suspended sediments, which was listed in a recent Department of Fisheries Oceans report. Sediments can have negative impacts on fish because it can clog their gills, reduce their oxygen consumption, and decrease the visibility around them. Because of the status of red side dace in Canada, it makes it hard to study them here. So our lab established an experimental population of dace taken from the states where they aren't endangered. My research aims to look at how suspended sediments affect individual and group schooling behavior of red side dace. So to do this, I will take groups of red side dace and swim them in a swim chamber shown in the picture with the red arrows. I will add a certain amount of sediment and increase the flow speed, and then I'll be able to look at metrics such as the spacing between the fish, as well as the orientation of the school. Our lab is also involved in different types of outreach. We have the Freshwater Restoration Ecology Center, FREC, in the bottom left, where we give tours to the public and to people of all ages to educate them on issues that the Great Lakes is facing, as well as on the species that we keep there, such as lake sturgeon and Chinook salmon, to name a few. We have recently partnered with Ripley's Aquarium in Toronto and have helped them set up part of their Great Lakes exhibit. We've actually donated some of our red side dace towards their exhibit in Toronto. And hopefully I'll be able to intern there in the near future to learn more about their conservation efforts. Additionally, we will be working with them again this summer to craft some science communication posts that they can put onto their social media pages. To sum up, urban threats are becoming more common. So this makes it especially important to look at stressors such as suspended sediments and how they affect aquatic species at risk, such as the red side dace. There need to be thresholds set to determine an appropriate level of sediment that's allowed into red side dace habitat. These thresholds can then be used by management and construction. For example, myself and multiple other graduate students at GLIER are involved in the Peel Region project, and this project is looking at how is assessing the urban threats to red side dace from a road widening construction project going on in this region. They're trying to determine the limits of sediment that should be allowed from this construction into the red side dace streams. These limits can then be used to identify critical habitat for red side dace, as well as for reintroduction purposes. Thank you.
Thank you, Madison. Again, I will appreciate it if all of you to fill up the feedback form for each of our presentation. And now I want to introduce our next presenter. It's Alison Lucas.
And part of the T-RUST program at Wayne State. So during my early academic career, I developed an interest in exploring the nexus of business and sustainability, and began by looking mainly at sustainable supply chains and the triple bottom line, a phrase coined by John Elkington in the early 90s. Through ongoing academic and professional experience, I came to better understand the important role of innovation and entrepreneurship in driving change. And Elkington has since gone on to petition for a recall of the phrase, citing its dilution into everyday jargon and its reduction into just another advanced accounting tool. As we collectively come to develop a more robust, comprehensive, and just idea of what sustainability is and requires, it's clear that we must start to think about system-level change. Inspired by the palpable spirit of innovation and a thriving entrepreneurial ecosystem that surrounds us here at Wayne State in the heart of Detroit, I've started to think critically about the role of entrepreneurship in that transformation, and in combating our world's most wicked problems. Sustainable entrepreneurship is the process of opportunity discovery, creation, evaluation, and exploitation, which leads to environmental, economic, and societal gain. Sustainable entrepreneurs, using their entrepreneurial spirit of creative destruction, are uniquely positioned to bring about radical innovation. They work to replace existing market structures and consumption processes with more environmentally and socially sustainable alternatives, and they provide value across a broad group of stakeholders. Despite being a relatively new field of study, research has produced a considerable amount of scholarship identifying what motivations, attitudes, and traits contribute to an individual's propensity towards sustainable entrepreneurship. But there's been little work done to understand how these critical success factors come to make entrepreneurial identities. As an early organizational communication scholar, I hope to explore what success means for sustainable entrepreneurs, how social discourse contributes to that understanding, and how that's incorporated into an entrepreneurial identity. This research will contribute to a growing body of evidence that supports sustainable entrepreneurship as a means to overcome traditional market failures, to reconcile development with sustainability, and to enact macro level change. It is my hope that by better understanding the determinants of success in sustainable entrepreneurship, we as scholars, educators, and practitioners will be better equipped to advocate for change, foster innovation, and facilitate learning amongst our peers, students, and our communities.
Thank you, Alison. And now you can see on the chat, there is a form for the feedback, specifically for Alison Lucas. I will be appreciated if you can fill out that form as well. Now I will introduce our next speaker, who is Lauren Weller. Thank you.
Hi everyone, my name is Lauren Weller and I'm an MSc Environmental Science student in the Healthy Headwaters Lab at GLIER. Today I'll be talking about carbon complexity as a restoration tool in agricultural landscapes. So organic matter acts as a bridge between terrestrial and aquatic environments and is fundamental to many biogeochemical processes in both environments. An intimate relationship thus exists between waterways and the lands that they drain, with landscape level characteristics such as underlying geology, soil type, hydrology, and land use greatly influencing both the quantity and quality of dissolved organic matter, or DOM, exported into freshwater ecosystems. So let's talk for a minute about organic matter, specifically dissolved organic matter. What we commonly refer to as DOM is in fact hundreds of mostly uncharacterized compounds varying from simple carbohydrates to highly complex aromatic molecules. Recently, we've come to understand these substances as recognizable compounds, albeit undergoing decomposition. Picture a pile of knitted items, each at different stages of being unraveled. Some may have originally been sweaters, but now only have arms or maybe the body section left, and some may have just the collar left. Chemical analysis can show us the wool blend, or in this case, the chemical composition, but establishing what item of clothing is being unraveled can be more of a challenge. Fortunately, DOM has certain fluorescent properties which allow us to study and understand this underlying complexity. Light at certain wavelengths is passed through a liquid sample to create what we call an excitation emission matrix, or EEM for short. EEMs can then be analyzed to identify specific components present in the DOM, and these components can then be uploaded to open source databases, thus allowing us to identify similar components and compare findings across studies. To extend the unraveling metaphor, using FDOM, we can sort unraveling knit items into piles according to type, i.e. sweater, blanket, hat, and thus identify what we're looking at. So why do we look at carbon complexity? Well, furthering the knit metaphor, imagine that the carbon itself is the yarn, and all the knots, beads, baubles are the nitrogen, phosphorus, iron, or other minerals. Well, you could measure the baubles. The yarn itself, or in this case, the carbon, is our best proxy for organic matter. In addition, carbon, specifically soil organic carbon, is considered a reliable indicator of soil health. This is because carbon plays a major role in binding and modulating nutrients within soils. Richer, more complex carbon stocks are associated with richer, more resilient soil ecosystems. And globally, we know we're facing a multitude of challenges with both freshwater quality and soil degradation. Investments in soil health, specifically investments in carbon, have been touted as the solution to both. But studies have traditionally been restricted to either the freshwater or soil science disciplines, with few multidisciplinary investigations at the soil-water interface. My research aims to characterize the dynamic relationship between soil and water DOM in agricultural landscapes by looking at the complexity of carbon in both environments. If you're interested in hearing more about what we have found so far, please follow myself or the lab on socials to tune into some of my upcoming conference presentations. But, spoiler alert, carbon has a lot of stories to tell us and may offer us a predictable way to infer restoration in agricultural landscapes. Thank you.
Thank you, Lauren. And again, there is a feedback part on the chat. I will be appreciated. Fill up the form for Lauren as well. Now I want to introduce our next presenter, who is Tilak Patel. Thank you.
Hi, everyone. My name is Tilak Patel. I'm in the GLIER lab at GLIER for environmental science, and this is my project titled Simulating Agricultural Stream Sediment Phosphorus Interactions. Hopefully I'll get more into what that means in the next slides. So some brief background before I get into my research aim. So we all know that Lake Erie commonly faces the issue of algal blooms, and these algal blooms are commonly caused or commonly fueled by one key nutrient, that is phosphorus. And so there's two main methods by which phosphorus reaches Lake Erie. One of them is by wastewater treatment plants, and the other one is by agricultural streams, which carries that phosphorus-rich fertilizer into Lake Erie by the streams. And so my project really focuses on that sediment-water interaction in the stream itself, where the sediment is not inert, but it actually reacts with the overlying water to either adsorb phosphorus into the sediment or desorb phosphorus from the sediment based on different environmental conditions. And so there's two different conditions or scenarios that I have here. So the first scenario is in turbid waters. So we have a good method for measuring phosphorus adsorption, desorption in more turbid waters, which is used by the batch method, a method that basically has centrifuge tubes, and you have your sediment in there. You have water and phosphorus concentrations, and you shake it for 24 hours. So that's a good kind of simulator for the first scenario, which is the turbid waters. But for the clearer waters where there's less turbidity, it's kind of more like a smooth flowing water stream with less sediment involved. Well, in that kind of scenario, we need a new method. And that's the one I created for method 2, which is called the mesocosm method, where basically you have a sediment core and it's got an overlying water layer, which is relatively clear from turbidity. And you've got a external reservoir, which feeds water into that core. And then the core brings it back via a pump. And so this is kind of better for simulating clear waters rather than the turbidity waters, which are the first method. And so now we have kind of two methods that are able to measure phosphorus adsorption, desorption. So we have a clear picture of when phosphorus is adsorbed and desorbed in two different scenarios. And so what I want to do is really target the vulnerable sites. So figure out which sites are more vulnerable to releasing phosphorus. And then hopefully we can utilize growing technology to be able to stop phosphorus from being released from those streams. And we can hopefully reduce phosphorus loads into Lake Erie. And that should translate to better food security and economy, because less algal blooms means that less species are dying due to their toxins and anoxia. And that should be better for healthier fisheries. And obviously that will improve economy. And with that, you also get clearer waters, because with less algal blooms, beaches are clear, and that is better for recreational use. So that is all really what my presentation is about. Thank you very much for listening. Hopefully this will give you a good overview on what my presentation is on, or what my project is on.
Thank you, Tilka. And again, I will be appreciated to fill up the feedback form for him, which is in the chat room. And now I want to welcome to our last speaker from Season 1, who is Samantha Connor, and I will be appreciated. Just be ready and good luck, Samantha, and this is for you. Go ahead.
Thank you. Hi, everyone. My name is Savannah, and I'm a master's student in the Wisner Lab at GLIER. Today, I'm going to be talking to you about the impact of agricultural drainage on environments within the Lake Erie Drainage Basin. Next slide, please. As we know, agriculture is extremely important in all of our lives, mainly because it contributes to our food security, but it's also really important in an environmental sense because it uses so much land to grow and produce these crops, which we then consume. One of the main issues that growers face today with their crops is excess water on their fields, which has resulted in tile drains becoming a very common practice. While effective in draining excess water, tile drains can facilitate the increased nutrient loading from agricultural fertilizer treatments, as well as providing microorganisms a possible pathway into nearby aquatic systems. With this in mind, my study aims to investigate environments which are connected through this agricultural drainage. These environments include soil, tile water, surface water, and sediment. Collecting and chemically analyzing samples from each environment after the application of an agricultural treatment will determine peak nutrient loading concentrations, and continued sampling throughout an entire growing season will determine what changes occur over time in each environment. I'm then going to investigate the level of microbial connectivity across terrestrial and aquatic environments. This can be achieved through DNA and RNA extractions and sequencing of the samples to see what microbes are specific to what environment, the functional capacity of communities, and finally to see whether microbes originating from an agricultural field are being transported through drainage into the connected environments. Next slide. To study these things, I'm going to be sampling two different field sites located in the Lake Erie Drainage Basin. One site is an isolated system, which you can see on the left. And this is where an agricultural field drains directly into a constructed pond. This will allow for agricultural specific nutrient and microbial communities to be determined. The second site I'm going to be sampling is more representative of a natural system. Along the agricultural transect, which is being sampled, there are additional human and environmental stressors. I'll be investigating if the same agriculturally related samples found in the isolated system are apparent in the natural system, and to what extent these responses are apparent both locally and downstream, with other stressors acting at the same time. Final slide. In terms of my projects linked to environmental sustainability, we know that agriculture links to other environments through transport pathways, and it's common for these transport pathways to be connected to larger aquatic bodies of water, for example, or drinking water sources, which means increased nutrient amounts and microorganisms specific or who thrive under certain agricultural practices can migrate away from the fields to these aquatic bodies and lead to degraded water quality, which may also have associated health consequences for human and habitats. Agricultural stress combined with additional human stressors can further add to this environmental degradation. Ultimately, if we know the extent to which agricultural practices have the potential to load nutrients and microbial communities and the ability of these to move downstream, we can better understand and mitigate through management practices the agricultural impact of water quality and health throughout an entire watershed. This will also help to maintain or improve the overall health of that watershed. Thanks, everyone.
Thank you, Savannah. Thank you so much. And we will now have a 10 minutes break. Please use this time to stretch your legs or refill your coffee. And please don't forget to fill out the survey from each speakers. And we will resume at 10:50 a.m.
Thank you for attending our first session of the day, and welcome to session number 2. We're going to get right into it with Chelsea Salter.
Okay, good morning, everyone. My name is Chelsea Salter. I'm a first-year master's student in Dr. Chris Weiser's lab at GLIER, and I will be presenting my research titled Tracking Microbial Gene Expression and Metabolites During Microsystem Degradation. Next slide, please. So harmful algal blooms are a persistent global problem that threaten the safety and utility of our freshwater resources. One of the main issues involving algal blooms is the release of harmful toxins from cyanobacteria, such as microcystin, which is a cyclic compound seen on the slide here. Being that it is a potent hepatotoxin, removing microcystin from water sources is critical for ensuring public and ecosystem health. Thus far, microbial degradation is the most promising prospective option for the removal of microcystins. However, a lack in understanding of the universal mechanism and how this degradation is occurring prevents the strategy from being utilized to eliminate the toxin from water bodies. My project aims to fill this knowledge gap by using multiple genomic tools to gain a comprehensive understanding of the degradation pathway occurring in Lake Erie, specifically in beach sand environments through using a combination of metataxonomics, metatranscriptomics, and metabolomics. My methodology will involve using both batch and column experiments in which the native microbial community within beach sand collected from Keeley Island will be exposed to lake water with the microcystin toxin. So first, my batch experiment, I will use various different variants of microcystin, such as MCLR, MCLA, and MCRR, to test the ability of the community to degrade variants that are prevalent in Lake Erie. The second part will involve my column experiment, with water containing varying concentrations of MCLR being passed through the columns that are filled with these sands over a 2-week period. The 3 main analyses I'll be performing are summarized along the left-hand side of my slide. The first being to perform metataxonomics, which will involve extracting the RNA from the sand, and then performing amplicon sequencing of the 16S rRNA gene to provide a taxonomic profile, and will be able to tell me which organisms are present and actively expressing RNA in my samples. RNA from the sand samples will also be used for metatranscriptomics, which will provide a functional profile and will be able to answer the question of which genes are actively being expressed and performing a function during the degradation process. And then lastly, metabolomics will be performed using liquid chromatography with 10 MS spectrometry to detect any microcystin degradation products within my water samples. And this will indicate where bonds are being cleaved and give critical insight into how microcystin toxin is being degraded. Next slide, please. The ultimate aim of my project is to work towards eliminating the toxicity that is caused by the presence of microcystin from harmful algal blooms, which are persistent and increasingly prevalent problem within Lake Erie. And so this work would therefore seek to improve the health and safety of the public and surrounding wildlife. The knowledge gained from this study aims to elucidate the microbial degradation pathway. So a future application could be that the community could be utilized in water treatment facilities to streamline the removal of microcystins from water bodies. And then additionally, by understanding which species are involved in microcystin degradation, this information could be used as a management tool to predict the susceptibility of lakes to persistent algal blooms based on microbial composition. And that's all I have for you. So thank you all for listening.
Thank you, Chelsea. We are now going to switch into our pre-recorded talks, and our first pre-recorded talk is from Matthew Day.
Chloride treatment and zinc oxide fire to have electricity generating clothing. Spider silk are among the strongest fibers known to mankind that are mainly composed of one or more proteins. The most investigated spider silk is dragline silk, which shows the most remarkable combination of strength and extensibility. The replicated protein of the dragline silk is the major contributing factor to unique physical properties, such as ability to supercontract when exposed to water. It also has been 3 times as true as RNA fibers and 5 times stronger by weight than silk, and its ability to undergo supercontraction. Shape memorial fabrics are made by dragline silk fiber, which responds to temperature, wind, and electricity simulation. The concept of shape memory fabric is new, and these fabrics were prepared by applying waterborne shape memory polymers using the shape memorial fabric that coated with the nanotechnology on the river edge could protect the city and any properties close to the river. When the river is overloaded, this fabric will open and extend the river pathway wall aid during the water flood. Thank you for listening to my presentation. You can contact me through my email address mentioned here.
Thank you, Gallery. Please, as a reminder, to fill out feedback forms, the link is in the chat. So we're going to switch gears here and switch back to our live lightning chats with our first presenter being Kira Hirshaw.
Okay. Hello, my name is Kira Harshaw, and I'm a master's student here at the University of Windsor's Great Lakes Institute for Environmental Research. If you live on or around Lake Erie, you're probably familiar with harmful algal blooms. Closed beaches, tainted water, and a putrid green tide are all symptoms of a now yearly summer overabundance in blue-green algae, including the microcystis species and the particularly notorious microcystin toxin they produce. Microcystins are well known and well studied, but they make up only a handful of compounds out of potentially hundreds produced by cyanobacteria during normal growth. And it's these enigmatic compounds that are the focus of my project. While researchers are still teasing apart the identities of these contaminants, it is becoming increasingly evident that these cyanobacterial metabolites detrimentally affect the health, growth, and reproduction of other aquatic phytoplankton, zooplankton, and plants. Of particular concern is the effect these compounds may have on fish. From eggs to larvae, prior research has found that these complex cyanobacterial mixtures have the potential to catastrophically disrupt proper growth and development in the early life stages of fish. But what about adults? Here in this burgeoning area of algal bloom research lies the focus of my project. What are the consequences of algal bloom exposure in adult fish, and how may fish populations be affected by algal blooms in the future? To begin to answer these questions, as well as further differentiate the toxic effects of microcystins from other algal compounds, my project will be comparing the cell-free filtrates of two Microcystis aeruginosa strains, one of which lacks the ability to produce microcystins, and their effects on the isolated immortalized cell lines of two freshwater fish species, walleye and rainbow trout. Cell line-based research offers an alternative to whole fish assays that will allow us to examine type-specific cell responses in cells from a range of different tissue types. Additionally, to get a better idea of the reproductive processes that could be affected, we'll be monitoring not only cell damage, but also how the expression of key reproductive genes changes following exposure. For many fish species, individuals will be forced to live, feed, and reproduce alongside this mosaic of algal metabolites. Disruption of reproduction and development can have serious consequences for these fish populations, especially those that may be vulnerable or facing other ecological pressures. With shifting climatic conditions and increasing anthropogenic stressors, these cyanobacterial blooms are becoming more pervasive, and as a result, we will see more and more fish species affected by these blooms. Understanding their effects beyond the influence of single isolated toxins is the key to improving predictions of the consequences of further expansion and in determining the steps that will need to be taken to protect our endangered species, mitigate algal bloom damages, and restore affected ecosystems. Thank you for listening.
Thank you, Kira. Our final presenter for our session, our second session of the day, is Alicia DiCarlo.
Hello, everybody. My name is Alicia DiCarlo, and I am co-supervised between the Weisner and the GLIER lab, where I am completing my PhD. So I was just going to talk about my very first publication for my PhD, which is going to lead into my following research, and this is going to discuss the microbial impact on phosphorus in sediment. So our first key aim was to show that microbial function has an influence on phosphorus mobility in freshwater ecosystems. To do so, I subsampled 3 streams that are agriculturally stressed. From these, I submitted a half to get gamma irradiated in order to tease out any biotic influence, as the gamma irradiation would kill any microbial activity. And then to ensure that this did not affect the physical characteristics of the sediment, we completed carbon analysis and particle sizing. So in that, we know that the irradiation did work, and we are looking in the living sediment only at impacts by the microbial activity. So what was great about this is that we found that there was a huge difference between microbial influenced sediments and the gamma irradiated. So we're able to say that microbes do influence the phosphorus cycle and therefore the mobility of phosphorus in sediments. So this links directly to environmental sustainability because we are able to see that microbes may mitigate phosphorus flux and be able to act as bioindicators of phosphorus cycling. We also need to look more into the mechanisms of microbial functioning in terms of the phosphorus cycling. And further research would have to include climate change, as our climate is warming. And so that's going to directly influence any microbes that are in the area for the phosphorus cycling and any other cycling that takes place. Further, we could look at different fertilizer practices as they also may influence microbial composition in these sites. Thank you.
Thank you, Alicia. So this concludes our second session. We're going to resume again at 11:30, which leaves us lots of time for everyone to give feedback to our presenters in this session. So just remember, each feedback form is for each individual presenter, and we highly appreciate the feedback, so don't forget.
Welcome back. We will now start our third session of the day with Smita Tyagi. Smita, we are ready when you are ready.
Thanks, Claire.
You're welcome.
Hello, everyone. I'm Smita Tyagi, PhD Earth Science student at School of the Environment, University of Windsor. Today, I'm going to talk about my research that I recently finished as MSc Earth Science graduate student. Next slide, please. The leftmost picture is a weed-free lawn. And in one of the pictures on right side, we see a tiny, pretty yellow flower dandelion, one of the most common weed found in our lawns. What do we do when we see a dandelion in our lawn? For me, I grab a ready-to-use herbicide spray and kill it before it overtakes the lawn. In my research, I studied iron-based herbicide and its effect on soil. In 2010, iron-based herbicides were introduced in Canada, and since then its uses has gone up many folds. Could this be a problem? Well, it depends on different perceptions. For me as a gardener, what I see is a black spot and the weed gone. But as environmental geoscientist, I see that I just induced iron in soil. Next slide, please. To investigate the changes in soil by iron-based herbicide, I used magnetic methods. Magnetic methods are based on magnetism. Magnetism is everywhere in nature in form of diamagnetism, paramagnetism, ferromagnetism, etc. The most common magnetic element that we come across in nature is iron. In soils, iron occurs in form of minerals like iron oxide, iron hydroxides, and sometimes even in metallic form. In magnetic methods, there are well-defined parameters, example, remnant magnetization, magnetic susceptibility, etc., which helps in identifying properties like the grain size, concentration of iron mineral in soil. Why it is important? Well, each property tells us the journey of that iron mineral. For example, iron mineral formed in waterlogged environment will have some particular magnetic properties, while iron mineral formed in dry environment will have different magnetic properties. If you look at the figure on bottom right, it shows magnetic properties, magnetic susceptibility profiles of two different types of soils called luvesolic and glossolic. In my research, I identified magnetic properties of different soil types. And with similar methodology, I identified magnetic signature of iron-based herbicide. My last step was to induce iron-based herbicide in soil samples and identify magnetic signature of treated soils. And finally, I compared the results. Next slide, please. Look at the figure on right-hand side. The change is evident. Similar changes were observed in other magnetic properties after treating with iron-based herbicides. The results of my study were able to answer the questions that I posed before setting my research objectives. Can we use magnetic methods to track usage and identify soil changes? And could iron-based herbicide affect or have affected previous or ongoing magnetic research studies on soils? Yes, we can use magnetic methods to identify changes, but the concentration of iron-based herbicides, which has only 4% of iron by weight, that is necessary to see these changes is unlikely to be used if the standard application instructions are followed. As of the previous studies and ongoing studies, my results suggest no significant effect because the usage of these herbicides started in 2010. Moreover, lawns are unlikely to be the sample sites for soil studies. And my next question was, could magnetic methods be used for monitoring to better project soils from pesticides, for more particularly for from herbicides for more sustainable soil protection and management? Yes, given the fact that magnetic methods are non-destructive, fairly fast, and not expensive as compared to the other methods, magnetic methods can be a diagnostic tool for such objectives. Thank you.
Thank you, Smita. Now we will switching to the pre-talk presentation and starting with Kate Akertor, and thank you.
My name is Kate Akertor. I'm a PhD student in the Department of Civil and Environmental Engineering at Wayne State University. I'm also a trainee with the T-RUST program at Wayne State University. The title of my research is The Role of Tree Canopy Cover in Water Quality Improvement Practices in Urban Areas. My key research aims are: 1, what are the quantifiable effects of tree canopy cover on stormwater quality and quantity, using the city of Detroit as a case study? 2, how do we increase tree canopy cover in areas lacking coverage in the city of Detroit? On this particular slide, you will see that I have utilized Jefferson Chalmers as my initial study area. And the reason is that I have decided to look at it at a smaller scale. I've decided to look at the effect of tree canopy cover at a smaller scale as against looking at it at a larger scale, which is the city of Detroit. Although looking at it at a larger scale is my long-term goal, but I felt it would enable me produce more accurate results if I look at how canopy cover is impacting quality and quantity of soil at a smaller scale. And what I have done is basically utilize the iTree suite of software. One of the software you will find in iTree suite of software is the iTree Canopy. And what this software does is that it helps you map out the cover classes. And when I say cover, what I'm talking about is the land usage within the defined study area, in this case Jefferson Chalmers, and it then generates a percentage land cover within the defined study area. This information generated is then used in your iTree Hydro software, and with other information like stream gauge and other soil parameters, you are able to generate quantifiable values. You are able to generate quantifiable effect of this particular cover class on the land, on the study area that you have chosen. This slide looks at how my research work is tied to environmental sustainability. And some of the things that urban tree canopy does is that it's known to regulate high temperature, which has been linked to increased precipitation. Secondly, increased infiltration is encouraged in soils that have trees. Thirdly, trees help reduce flooding. And that is my research. Thank you very much.
Thank you, Kate. And I appreciate it if you go and fill out the form feedback for the Smita and Kate. And now we'll go and start seeing the pre-talk presentation for Jess Robinson.
My name is Jessica Robson, and I am a master's student over at GLIER, working with both Ken Drillard and Catherine Febria. And today I'd like to talk to you about benthic macroinvertebrate communities of Detroit River wetlands. So we're all pretty familiar here with where the Detroit River is. It's a connecting channel that cuts between Windsor and Detroit. And on the Canadian side, there are 5 wetlands that are marked here in purple. And 2 of these wetlands, Turkey Creek and River Canard, are adjacent to tributaries. And so we want to ask the questions, do benthic invertebrate community assemblages vary between these 5 wetlands? And for the 2 wetlands that have tributaries, we wanted to know, was there any sort of internal variation with respect to the tributary? So they were separated by upstream to downstream, and we also sampled in the trib. And to answer the question of whether or not there was variation between the wetlands, we had taken a total of 50 samples, 10 per wetland, using petite ponar grabs. Back in the lab, we identified about 56 different taxa to make these data sets. And for variation between the 5 wetlands, there's somewhat of a difference, but I really wouldn't say that there is a significant one. And with respects to the tributary, in River Canard, there was really no difference whether we sampled upstream, downstream, or in the tributary. However, for Turkey Creek, we have a little bit of a different story here. So we have the wetland sites all clumping together here, but the tributary is really its own thing. And the link to environmental sustainability is that benthic macroinvertebrates are often used as bioindicators, and they are part of biomonitoring programs. And the Detroit River is a Great Lakes area of concern. And one of the impairments listed is the degradation to benthic communities. And this listing mostly has to do with sediment contamination problems, which have been improving. But I think if we want to better design biomonitoring programs for their long-term stability, we would greatly benefit from understanding more about the benthic macroinvertebrate communities. And with that, I would like to thank you for joining us here today.
Thank you, Jess. And now we will go on to start our last pre-talk speaker, who is the Yu-Ting Chen.
Good morning, everyone. My name is Yu-Ting Chen, and I'm a PhD student working with Dr. Crossman and Dr. Weisner at GLIER. I'm studying nutrient transport and downstream ecological responses across river-lake interface. And the topic that I'm going to talk about today is application of real-time technology on HEPS control. The binational efforts made by Great Lake Water Quality Agreement that aims to control eutrophication and nutrient pollutants have successfully reduced total phosphorus loading into Great Lakes. However, despite the improvement of water quality in the past 2 decades, we are seeing an increasing frequency and extent of harmful algal blooms, especially in nearshore areas in the Western Lake Erie Basin. The resurgence of HABs is likely to be associated with changes in nutrient fractions. For example, rate of soluble reactive phosphorus to total phosphorus, water temperature, flow regime, water clarity, and internal nutrient loading. And the nature of these stresses is highly spatial-temporally variable, which is difficult to capture through conventional sampling on bi-weekly or monthly basis. As a result, to understand the relationship between these stresses and rapid biogeochemical cycles within the river-lake corridor, our study aims to use real-time technology to bridge the data gaps and identify current drivers of HABs within the Lake Erie shoreline. We choose one of the most nutrient polluted streams in Ontario as our study site, Swimming Creek, because it is considered as a priority target and mitigation plan by the local authorities. Data we collected from upstream to downstream and lake shoreline through the sensors listed here, as well as analysis from the soil, eDNA, and RNA samples along the river transects, will provide us a holistic view on bloom control and developing models for bloom predictions. Based on the UN definition, environmental sustainability can be defined as maintaining natural resources and meeting the needs of present without compromising the need of future generations. Our study falls within this scope because, from the nutrient pollution perspective, nutrients such as phosphorus is a limited natural resource which supplies a large part of global food production. However, the excessive amount of nutrients can lead to eutrophication, which deteriorates water quality and impairs freshwater ecosystem at present and in the future through impacts of legacy nutrients. With the use of real-time sensors and high-frequency data, it will help us better understand the causes of current HABs and provide insight into nutrient transport during extreme events, preparing for projected climate scenarios in the future.
Thank you, Yuting. And please don't forget to fill out the feedback form for Jess and Yuting. And now I want to introduce our last speaker from Session 3, who is Ryan Graham. Good luck, Ryan.
Thank you, Gallery.
You're welcome.
Awesome. So yeah, as Gallery mentioned, my name is Ryan Graham. I'm working out of the Healthy Headwaters Lab at GLIER in the University of Windsor. And this is my project for my master's in environmental science. So my project is called The Characterization of Riparian Vegetation and Agricultural Drains Impacted by Phragmites australis. And I think I'm just going to ask for the next slide. Awesome. So biodiversity across the globe is declining rapidly. And according to scientists, 1 million plant and animal species are at risk of extinction within the next coming decades. The rapid expansion of agriculture and the resulting habitat loss can be tied to 70% of that total biodiversity loss globally. Historical wetlands, streams, rivers, and other aquatic environments are altered into massive agricultural fields with heavily managed drainage systems. While their main purpose, drainage systems, is to remove excess water from fields, these drainage systems are also responsible for a list of ecosystem services that was once provided by a natural stream, including nutrient retention, sediment and flood control, habitat, and many more. While these drains can provide a full list of services that I just listed for you, many drainage systems are plagued with invasive species of ryegrass, Phragmites australis, a plant that grows quickly, transforms these ecosystems, and restricts many of the ecosystems that I just listed for you. Agricultural drainage is a very heavily studied topic, and as you can see from today's presentations, there's a lot of projects that are still looking into specific aspects of drainage. Our understanding on these systems has expanded greatly, and research has shown that vegetational communities within these drainage systems have an important role in some of the services that they provide. For example, specific plants are important for bank stabilization or sediment control, or specific root systems from plants can provide better habitat for microbes that influence phosphorus cycling, like Alicia was speaking about earlier. This brings me to my research. While we know vegetation is important for these ecosystems, there are a few studies trying to investigate the specific vegetational communities within drains, how these communities impact local biodiversity, and how these communities are also impacted by Phragmites. Next slide, please. My research will attempt to investigate vegetational biodiversity, the impact from Phragmites and Phragmites management regimes, as well as investigate the functional traits with the communities I have that I will be finding. I will be conducting a seasonal biodiversity survey within drainage ditches across Windsor-Essex County, an area that I'm sure many of you are aware is heavily impacted by agricultural domination. The vegetational surveys will be conducted in the spring months of April and May, as well as the late summer months of August into September. There's no designated methodology for sampling drainage ditches, but the figure in the middle, figure 2, shows a proposed methodology. The survey will be a combination of transects and quadrats, with quadrats being randomly placed on key ditch habitats, which we've identified as lower and upper ditch habitat. Then this will be replicated at random intervals along the ditch to get a full representative of the entire ditch system. We used standard protocols like OSAP, OBBN, which are the Ontario Stream Assessment Protocol and Ontario Biomonitoring Network. We use those, and we're going to be collecting a presence-absence data set for these drainage systems, and will be one of the only studies that has done so in Southern Ontario. Alongside this, we will also attempt to identify sites with differing levels of Phragmites impact and levels of Phragmites management, including sites that are pristine and have never been managed to sites that need to be managed yearly for Phragmites. From this data set, I will also be conducting a functional trait analysis. I'll be using the species list that I find, and I'll be conducting a literature review to identify if any of the plants that we found in my survey are providing services to the drain, such as bank stabilization or nutrient retention. This part of my project will look into possible patterns that we find within the data, and will also be used to better understand some of the possible relationships we find for these communities. Also, will be groundwork for future research. Lastly, this data will be explored using traditional biodiversity indexes such as species richness or Shannon-Weaver, and further explored using RStudio. Next slide, please. So, with biodiversity in decline, I think it's extremely important to continue to study drainage systems, agricultural drainage systems, and identify the contribution to local biodiversity, mainly because in some agricultural systems, this may be the only place that biodiversity can thrive. My research will hopefully add to the field of knowledge surrounding Phragmites management, and also create a better understanding on what vegetational communities remain in these ecosystems. So thank you guys for listening to my presentation. As you probably have kind of clued in, April is here, and I'm planning on doing my surveys very, very soon, within the coming weeks. So if you are good with plant identification and would not be bothered by an email list, I want to create something where I can reach out when I inevitably find my 8th grass species that I can't identify. So feedback with that and any information for identification would be greatly appreciated. So thank you so much for listening.
Thank you, Ryan. So wrapping up our showcase today, I would like to leave everyone with a quote here. Every brilliant experiment, like every great work of art, starts with an act of an imagination, by Jonah Leher. I think we've seen that throughout all of our pre-recorded and live presentations today. On behalf of all graduate students and faculty from T-RUST and GLIER programs, thank you for joining us today. And a final reminder to fill out those feedback surveys for a chance to win prizes. Thank you, miigwech, and stay safe.